Hello, everyone. Greetings from the Society for Laboratory Automation and Screening meeting 2026. I'm Uduak Thomas, Senior Editor with Jen, and I'm here with.
Hi, I'm Julianna LeMieux, Deputy Editor-in-Chief at Jen. How you doing?
Hey, good to chat with you. I'm excited to do this. So, we're really excited. For the first time this year, we're doing a special issue newsletter for SLAS. And so, we just wanted to take a few moments from the meeting. We've both been here about a day and a half, I think, fair to
Yeah.
say. And so, we just wanted to catch up and talk a little bit about some of the things that we're seeing at this year's meeting. So kind of first thing, I wanted to get your overall impressions. What are you seeing? How are you feeling? What do we think?
Yeah, so this is my second time at SLAS. What I think same
Also,
for
second for
you?
me as well.
Okay,
Yeah, I
yeah.
believe so.
So I
Yeah.
was here last year, but it was in San Diego, so way warmer than
Yes.
Boston.
Very different temperature.
Um, but I would say one thing that's similar to last year is like the energy on the floor.
Hmm.
I mean, there's just like SLAS is one of those meetings where, there's lots of great sessions as well,
Hmm.
but like there's the floor is just full of energy and there's
Hmm.
tons of um big booths. And I think I one thing that we had talked about, I think, was
Hmm.
how there's a lot of booths that I've never even seen before, new
Absolutely.
names.
Yeah, yeah.
So
No, I agree. I was here 2 years ago when it was in Boston. I believe in this same center, if I'm not mistaken. And some of the big names are back this year. We were talking earlier about some of the
mhm.
biggest booths we've seen. Some of these are quite massive. And
Like bigger than my apartment.
Yes, huge with almost like stations. It's really quite impressive. And some of them I think are probably names a lot of our audience will recognize. Hamilton has one of the largest booths
Mhm.
here. We've also got Nexus XP, Tecan, Ginkgo, Thermo, Biocero, a newer company that well new to me, I think, Automata, SPT Lab Tech is here. Another company called Synomics, Revity, another really big name and another company, Bionex Solutions. That's by no means the all the companies here. We'll talk a little bit more, I think, about some of the others that I hear, but those are some of the ones with the biggest booths that we've seen at the meeting so far. But I agree, a lot of energy. And I think we were both saying it does feel like there might be more, like you just said, more booths
Yeah.
than when I was here and when you were here last year. So,
Well, and actually, can we
yeah,
just pause on Ginkgo for a second?
yeah, absolutely.
Because Ginkgo was here last year for I think the first time. And, really, that's when they were, I think, starting the evolution from like their kind of old model of the
Mhm.
biofoundry to new, automation
Mhm.
providers.
Mhm.
And they're actually have a huge presence here this year.
They
So
really do. Yeah.
that's kind of an interesting just change for
Yeah.
that company
Yeah.
that and I know that you went over to see
I
the
did.
automation center, right?
Yes, on Sunday night they had a little gathering. So that was actually my first day here at their office here in Boston. And so I had a chance to go over there, see the space, got a little tour of the lab, and actually saw some of the machines and equipment that they have, and got to ask some questions of some folks on staff. So it was a very interesting experience and very fun to see. I got some cool pictures and videos of things zipping around. So.
Yeah,
It
actually I
was
saw
very
Jason
exciting.
Kelly posted on LinkedIn kind of a teaser and he had showed video of the center as well.
Yeah,
On Super Bowl Sunday too. You
I
went over
know.
to that.
Yeah, that was
Yeah.
a Super Bowl Sunday in the middle of one of the coldest
Yeah.
winters in record. So, but definitely fun. Should we talk about some of the talks that are going on? There've
Yeah.
been sort of a mix of talks on the floor and some off. So let's start with maybe a few of the ones
Oh
that
yeah.
were on the floor.
Oh yeah,
Yeah.
sure. Yeah.
I mean, actually, why don't I go
Okay,
first?
sure.
Yeah, and one of the companies that I had a chance to see is called Brainstorm Therapeutics. And something we were talking about earlier. There's a fair amount of presentations on organoids
Yes.
at this particular meeting, and this particular company was super interesting. So the talk was done by the company's co-founder and CSO, Dr. Maya Goslia. I hope I'm saying her last name correctly. And she was talking about the company's work on developing these really, really great brain organoid cultures that they use for screening different drugs in the context of well, we're working on the brain, so diseases related to the brain and CNS. And so specifically, she talked a little bit about the the development process of the organoids and the accurate results they get from using these for screen in different sort of drug discovery initiatives. And so she talked a little bit about some of the lines they used for testing for drugs in conditions such as Rett syndrome and
Yeah.
Parkinson's disease. And as a matter of fact, something super interesting about what they're doing is using their organoids, they were able to identify a drug that was actually initially, I think it might have been used for uh, I forget the particular disease, and I don't want to misquote it, but they basically repurposed the drug for use in Rett syndrome, and just based on the efficacy data from using organoids, they were able to get IND approval
Hmm.
to move forward with this particular drug in, I believe it's phase 2, without any requiring any additional animal data, which I
Yeah.
thought was really, really cool.
Which is
Um,
happening more and more now. Yeah.
yeah, yeah,
Yeah.
and I do recall. I apologize. I misspoke. I stumbled a bit earlier. The drug that they're repurposing was initially for Alzheimer's disease,
Okay.
and that's now being repurposed for Rett syndrome. The FDA has cleared it to go into clinical trials, which is pretty exciting to see how organoids have really come a long way.
Yeah.
And there's a lot of great conversation
Well,
around
and also
organoids.
married together with the automation here, they can just
Exactly.
screen so much more quickly and through so many more drugs and things like that. So yeah.
Yeah, yeah. So that's definitely a company that I think I would love to see how things work out for them. But maybe we can talk a little bit about some of the other talks on the floor.
Yeah, I mean, one thing that's kind of neat in these big booths, at least in the OpenTrons booth, is that they're doing these like 15-minute talks. And so
Mhm.
you're just kind of walking around and you can just, jump into a talk instead of like going all the way to the sessions, such a walk. So I just jumped into a talk where Plasmidsaurus, which is a company that's really gained a lot of, momentum
Mhm.
in the last year,
Mhm.
explained how in there they do, DNA sequencing and also RNA sequencing, and how they've kind of how they rely on the Opentrons robots to help them do it faster, cheaper, more higher throughput.
Mhm.
So that was kind of an interesting partnership between them. And then also one thing, and not surprisingly, I'm kind of drawn to the omics talks here at SLAS. And so that was one, at least DNA sequencing.
Mhm.
But another session that I went to yesterday morning, Molly He, the CEO of Element,
Mhm.
was the moderator of the session. It was all omics. And so, and just, I mean, I know that I write about omics a lot, but looking at it through the lens of how automation is
Mhm.
really um changing,
Mhm.
some of the game for, I mean, I hate that term game changing, but anyway, changing some of the workflows of
Mhm.
how omics is being done. So there were several amazing talks in that session. So Noor Ahmed spoke. He's a postdoc in Emma
Mhm.
Lundberg's lab. And he started by talking about, virtual cells. And it's like
Mhm.
such a buzzword right now. And we have lots of cell atlases now with, Illumina has one, Arc has one, BioHub has one. There's lots of millions to billions of cell atlases. But they are really focusing on not just building a cell atlas, but doing like perturbational
Mhm.
biology
Mhm.
to build cell atlases.
Mhm.
And Perturb-seq is so expensive. Cell Paint is great, but you
Mhm.
have to screen through millions of drugs. So they're using a new technique called optical phenotyping,
Mhm.
which is faster, allows them to do more. And then he spoke then about how they're utilizing the robotics in order to kind of make that happen. So that was super interesting.
Mhm. Mhm.
Also, I know I can't like go into detail of all these, but Gail Marshall spoke from Medicine's Discovery Catapult.
Mhm.
So totally different topic. I really like this session. There were like three totally different talks, and they're trying to develop biomarker strategies for Huntington's disease. And they're doing tons of omics on
Mhm.
patients of Huntington's disease samples of CSF and plasma.
Mhm.
And so they were using Luminex and Olink for proteomics. They were
Mhm.
using mass spec. They did metabolomics, lipidomics, and they use the geomics for transcriptomics. And so they have identified novel biomarkers for
Mhm.
Huntington's that they're, working in animal models and these patient samples. So that was super exciting, I thought.
Interesting. Yeah.
And then the last talk was by somebody from Atrandi
Mhm.
Biosciences, a company that's been around for about a decade. They're in Lithuania,
Mhm.
but they're right now starting to talk about a new single cell platform, like scalable single cell for co-profiling of transcriptome and genotype. And so they have these things called SPC, semi-permeable capsules. And this is their technology and combining that with liquid handling technologies to do like high throughput single cell screening. So I'm actually planning to talk to them. I think they're going to be at AGBT in 2 weeks. So I want to catch up more on the technology and how it works and these capsules, which seemed really interesting. And he said that they're going to be launching that product like it's a while out. Like they're still in early access, maybe 2027. Yeah. So some really cool, and there's more omics today as well. So I guess just sticking to what we know there.
No, absolutely. I'm really interested. A lot of really great omics talks. And I mean, to be clear, that is not the only thing
No,
going
no.
on at SLA. There's a lot more. But I'm gonna talk a little bit about the keynote. You know, one of the things that we've talked about, so this was the first keynote sort of kicking off the entire meeting this year. One of the things we've talked about quite a bit just at Gen, but also even on the floor here, is how much automation just permeates so many different areas that we cover and that so many of our colleagues in the life sciences cover. So it's interesting to see just the diversity of talks that do happen at this meeting. And tying that to the keynote, it was by a company called Accent Therapeutics. Specifically, it was given by the Chief Scientific Officer, Dr. Serena Silver. And it was what really struck me, and it was a very interesting conversation. I'll tell you a little bit more about it, but it was very, very biology forward. And again, it just ties back to just how much automation is essential for so much of the things we write about and the work that we cover. So specifically, she was talking about the work a company has done in developing an inhibitor for a particular kinesin motor protein called KIF18A. This particular protein, there's all kinds of kinesins in the body. This particular protein shows up. It's particularly helpful for cells that are dealing with with different types of genomic instability. So chromosomal rearrangements, issues with defective DNA repair. So they need a little bit of help to come along and get them during cell division. And that's where KIF18A kind of comes in. And so what they have found, and she presented, and I do want to say their work builds on a lot of research that others have done on this particular protein. What they found, what they were going for was recognizing that this particular protein is active in cancer cells that have genomic instability. The idea was to develop an inhibitor for this particular protein, and then that would work well for patients who have cancers that have cells with a high degree of instability. So an example would be ovarian cancer, which was, in some of the results she showed during her presentation, were tests done in both ovarian cancer cell lines and mouse models and xenografts as well. But these chromosomally unstable cells also show up in things like head and neck cancers, lung cancer, and I believe breast cancer as well. And so a lot of the presentation was a lot of data, just kind of showing just it's kind of building the story around why KIF18A is so important as a potential oncology target. And then she also went into a lot of detail about how they identified a particular small molecule that they were going to use and just the ways they optimize it, including at one point adding a silicone atom at one point, looking at the binding pocket and kind of getting the crystal structure of that and the ways they changed it. Medicinal chemists on the team changed it just to basically optimize it to bind to this KIF18 protein and inhibit it. And then she showed results of testing, as I mentioned, in cell lines and mouse models, and also so in patient-derived xenografts, just showing that when this inhibitor was used in types of cancer cells that are chromosomally unstable, it worked. Cells ultimately committed apoptosis or went through the apoptosis process. Basically, cell division stopped and they went through apoptosis. And then they tested it in normal cells as well as cancer cells that don't have the chromosomal instability issue. And those cells were largely unaffected. They were able to continue doing their cell division because in those instances, KIF18A was not as necessary or was not a necessity since they weren't dealing with the instability. So it's a very targeted cancer therapy. And it was very interesting. The results seem really good. The particular inhibitor that they have developed, I believe it's called ATX 209 or something along those lines. I want to be really careful. I'm sorry, 295, not 209. I want to be careful about misstating things. Um, it's doing real well. They've dosed their first patient, so it is in clinical trials, and it's really interesting to see the kinds of science that folks are doing, and you know, kind of the results that are coming out of the field, just sort of more generally. So it was a very biology forward talk for an automation conference, but again, I think it just underscores how much automation just permeates so much of the research that we do in biology, life sciences, drug discovery, and so on. So yeah, I will definitely we're going to be writing about a lot of the stuff that we're talking about, and hopefully, if you have not signed up for our newsletter, you do so, and you get to see all of the content in a nice, concise email. But if not, definitely feel free to check out the website for all of the coverage on SLAS that we're doing. Well, we have a couple of minutes. Anything else going on that you wanted to talk about?
The only other thing is I mean, I spent a little bit of time yesterday just walking around the floor. I went to talk to a Trandy more. That was that company I mentioned earlier. I did speak to, this is a company that they're kind of like part of Yokogawa, and they have a platform called FlowCam. And I've bumped into the creator of this instrument before at BPI and bioprocessing because they use images to identify or, allow you to visualize contaminants in like injectable drugs. But here, actually, I mean, it still does that, but they're using it more to look at organoids. So we were talking about how organoids are so hot. So that was kind of interesting to see like that this machine is being used for a totally different purpose. But it's like, yeah, visualizing all different organoids in different, you can and perturbations and things like that. And then I also stopped by this booth because they said something like organelles in a tube or something like that. And I was like, wait, what do you do? So they make organelles right now. They just have lysosomes available. But yeah, so you can purchase organelles from them. So it's like, it's sometimes just going to a booth and saying, what are you doing? Is really interesting because there's a lot of people doing a lot of different things.
Yeah, absolutely. Absolutely. I did want to highlight one more thing. There's also, I mean, as with conferences like these, there are a lot of new products, new partnerships that are being
Yeah.
announced. Unsurprisingly, there's a lot of conversation around AI and the integration with automation. Both in terms of both products and partnerships, or just conversations around that being sort of the future of lab automation and what that might look like, and what companies are sort of trying to take those first steps into that space. We covered a partnership between OpenTrons and NVIDIA in that regard. And there's just a lot more. I think it would be really, if we started trying to list all the possible AI automation integrations or conversations that are happening at this meeting, I think we would end up taking way more time than we have for this video. But we're delighted. It's been a really great conference, I think.
Yeah.
And I mean, I'm sure I'm not sure where it is next year, but I'm sure wherever it is, we'll be there.
Yeah, I hope so. Yeah.
And yeah,
Automation is not slowing down
it
at
is
all.
not.
So
It is
yeah.
not at all, at all. But thank you so much for catching up with
Yeah,
me. This
thanks,
was
Uduak.
great.
Yeah, it's
Yeah,
fun.
we'll hopefully we'll do this again next year.
Okay.
Okay.
All right. Thank you.
Bye.
Bye.